Thank you so much. Now, alright. So, the basic explanatory paradigm is that of Z-scheme and Kok-Joliot cycle. If you are from the biochemistry side, these terms will be familiar to you. So, I'll directly get into the talk. Okay, so there you see on the left side is the famous Emerson experiment that determined the course of photosynthesis research. Basically, what they did is they illuminated the chloroplasts at two different wavelengths, standing for the two photosystems, and then they saw that the yield of oxygen evolution was much higher when you combine both wavelengths. That's what is shown here. And then Kok-Joliot cycle is here, wherein you see a four cycle periodicity. So they said that 4 electrons constitute to 2 water molecules breaking to give 1 molecule of oxygen evolution at one point. So this defines the crux of photosynthesis research. So you have the electron transport chain and the proton pumping, for which Peter Mitchell got the Nobel, and the rotary ATP synthesis, for which Paul Boyle got the Nobel. All these things I am going to challenge in this talk. Alright? So the first thing is the serial connectivity of photosystem 1 and photosystem 2. It's very elementary. Now you've got two systems. When you get connected, whether it is fluid mechanics or whether it is electrical electronic system, you get a greater flow in a parallel system, not in series. So the first mistake is there. This is anything that even a 10th standard or a 12th standard student with mathematics or fluid mechanics can understand. So if you get more yield with photosystem 1 and 2 combined, they have to work in parallel. They cannot be connected serially. So there goes the whole community gone wrong. Alright? And the second part is, some people are from cyanobacteria. In cyanobacteria, you have both photosynthesis and respiration going side by side. Now, imagine this. Photosynthesis is flow of electrons from water to NADPH, whereas respiration is from oxygen to water. So, how can they work side by side? For example, cytochrome C in respiration it has to take from 1 to and in the photosynthesis it has to go opposite. Most important is the cell volume of a cyanobacteria. A cyanobacteria is sub-micron dimension. It works around pH 7 and 8. You have the water content. You try to calculate the number of protons. It comes less than 1. What is Peter Mitchell's theory? Protons are pumped out. There are no protons to pump. Less than 1. So this is something a 12th class person can calculate, and this was given a Nobel Prize. And this is published. So this paradigm is going to go out. And most importantly, electrochemical system, when you have maintained it at a particular potential, positive, negative, the electrons or the ions are supposed to move in a particular way. Here they go all over. It's a deterministic mandate because some people wish it, but it cannot work in realistic environment. So this is on multiple level. So now if you know the basic chemistry of it, there are mobile electron transporters like plastoquinone and plastocyanine. Plastoquinone is isoprenoid with quinone, and plastocyanine is a protein, copper containing protein. Now you see, they knocked out plastocyanine in 1995 or 97, and they found that the plant works quite perfect. So plant in the Z-scheme, plastocyanine is the electron transfer agent from cytochrome B6F to photosystem 1. So how can that scheme work? This is completely avoidable, and also its structure is not concerned. And also you see plastoquinone, photosystem 2 to cytochrome B6F, they have to jump barriers. It cannot. These are all unrealistic ideas. And then it is supposed to do quantum These light harvesting complexes are supposed to harness light and give excitons. How? How does it go? It's an amorphous system. It cannot. So basically this I already said, the electrons are going here and there, the protons are going here and there. There is no abiding to any electrochemical laws. And then very clearly you can see Z-scheme. You take 4 photons of 680 nanometers and 4 photons of 700 nanometers. That is the, as per the theory, and calculate how much energy comes to. You see the standard textbook equation. The amount of energy input is much lesser than the amount of energy needed for the reaction. Thermodynamically not viable. So at multiple level, this scheme has flaws. It is not sustainable. And I have shown in great detail, I have analyzed each step. Here what I am introducing is a new concept called murburn concept. This is my baby. Now what I am saying in this is, classical enzymes work with Fischer's lock and key kind of mechanism, right? You have enzyme active site, the substrate binds, and then you have electron transfers or catalysis. Here, what I say is, let's imagine beyond that. Let's imagine that there are some enzymes that work with diffusible reactive species. For example, oxygen can get reduced to superoxide and they can move electrons from one point to another. You don't need deterministic electron transport chains. So now you must remember that photosynthesis is a light presence. It is done in the presence of light. Chloroplast cannot avoid oxygen. So here in my theory, you have all these photosystem, cytochrome P6F, NDH, NADPH, dehydrogenase, all these, the protein content that hangs outside the membrane, that part has ADP binding. And when the reactive oxygen species is produced because of effective charge separation occurring due to light activity, the oxygen-centered radicals that are produced that attack phosphate and ADP to make ATP. And when these reactive oxygen species react among themselves, you get oxygen evolution. And the electrons that are liberated, they are harvested directly by ferredoxin and then NADPH is made. Now, this is a highly decentralized, completely different idea. That is, it's murburn. Mur is closed. Burn is an oxidative process that might involve chaotic stochastic phenomena involving diffusible radical species. So that's what you see here. This is the paradigm. So you have ATP. You see the equations are all bimolecular and all of them have calculated the thermodynamic yields. They are all bimolecular, simple reactions. They make ATP, oxygen evolution, all that. This I have shown through multiple papers from 2006 to 2020 that you can get oxygen superoxide equilibrium relaying electrons. You don't need deterministic electron transport chain. This is how something can evolve. You cannot have deterministic processes. Now here, I have also shown that if you just take ADP and phosphate and just add superoxide, you get ATP. It's a very simple system. This thing was missed by people because they just thought superoxide is toxic. It's a reactive oxygen species. It's bad. It's bad. But then that's the reason why we need oxygen, why oxygen has become such a big player in life. So it's not just me who has done this. In 1987, some other researchers have done it with xanthine oxidase, which shows ATP production. So you see the structural components of chloroplast. See the proteins, how beautifully they support the new theory. You see, the proteins are highly porous. The chlorophylls and carotenoids, they are all scattered all around the place. There is not covalent binding, there is no deterministic light harvesting complexes placed here and there in a particular bowl or anything like that. It's a stochastic phenomenon. It's a chaos-ridden, chance-based phenomenon. not a deterministic phenomenon. Completely different outlook. So basically, multiple low affinity ADP binding sites on the parts of the proteins that extrude beyond the membrane, and you can see the other supporting evidence is multiple diverse types of molecules can give and take electrons from both photosystem 1 and photosystem 2. If electron transfers were to be very active site based and all that, you cannot get such kind of outcomes. So basically, bicarbonate is also now a very capable donor. It's not just water breaking. Bicarbonate has got lesser requirement to break than water. You see that the calculations are very clear, and so here it explains. The classical Z-scheme cannot explain how when you give bicarbonate you get more oxygen. The new theory can explain. Alright, so now basically, so flawed foundations, multiple Nobel Prizes have been given. 2 are out already. Paul Weyer's rotary ATP synthesis. There is no way rotary ATP synthesis can work. Okay, so I have critically analyzed the structures. These papers are already published. If you take a fruit, you can put it in a mixie and you can get juice and pulp. Alright, but you cannot take juice and pulp and put it in the same mixie and get fruit. What the classical theory says is, of course we can make fruit in nature, but it is made through another thermodynamically facile process. That's what I have detailed here. So these things will have to go, and Rudolf Marcus outer sphere electron transfer, I have no questions again about it, but its relevance here is very limited. And also the Eisenhower structures on bacterial photosystems, they were very ordered. Bacterial systems are very ordered, but not plant systems. Alright, so these are my collaborators. Basically, the conclusion is I'm a theoreticist and I'm a biochemist. Basically, so my work, I have not shown you the major experimental data or the in silico explorations. What I've shown you is a snapshot of how things are going to change. Alright, here what I'm saying is let's discourage Z-scheme, ETC, Kok-Joliot cycle, Q cycle, chemiosmosis, proton motive force, rotary ATP synthesis, all these terms, these are untenable. It doesn't matter how many people believe in it, how many years have gone by, 80 years of misdirected research, high time that we have to change. So it doesn't require multimolecular complexing, plastoquinone going about in a definite way, plastocyanin going about in a definite way. It's a very simple bimolecular chaos-ridden process that can spontaneously work. This is my mentor Lowell, and this is Nikolai Biden from Russian Academy of Science, and this is Yan Yu of Chinese Academy of Science, my collaborators, and these are my students. So with that, I have taken about 9 minutes. I just wanted to show these are the extra calculations that I have made in each single step. Can you imagine? Several dozens of steps are needed to make one molecule of NADPH. It's impossible. People have gone on a wild goose chase, and the classical Jagendorf experiment, which was taken as support, it's a very simple experiment. What they did is they took mitochondria and pH 4, they equilibrated it, and then they exposed it to pH 8, and then saw ATP. The equation is very simple: ADP plus phosphate plus H plus gives ATP plus water. You will get ATP. This is equilibrium driven ATP synthesis. This has nothing to do with physiology. Nothing at all to do with physiology. This was taken as a support for chemiosmosis. It's because it cannot be pH 4. They found in 1970s itself, all of them work around neutral pH. There is no pH gradient or anything. So, but then they gave all. This is the Racker's experiment. Rhodopsin was taken as a proton pump. Rhodopsin is actually carotenoid. It produces ROS. So it is a misinterpretation again. So this is the different views of my own theory. Basically, Why do we need then photosystem? Photosystem does effective charge separation. Light harvesting complexes all tap light, we give raws, but then they are not directional. They produce raws and then go back. Effective charge separation is only noted with photosystems. So now this is a completely new paradigm in photosynthesis chemistry. I thank you, sir, the chair, for giving me the 10 minutes. I thank Dr. Menon for giving me this opportunity to speak, and I thank you all for this patient hearing. Thank you so much. Thank you.
Thank you, Manoj. Really extending presentation. So we got a overlook on the photosystems, the way we study, what we study. I think Manoj is trying to compare with the photocell system.
Basically, what I'm trying to do is, sir, I'm trying to completely revamp cellular biochemistry, cellular biophysics, and cellular physiology. This is the first one in photosynthesis. I've completely revamped respiration, homeostasis, thermogenesis, electrophysiology, you name it. How you see the eye, all that, how nerves conduct, everything. Cellular physiology is going to change. And I hope this is Swadeshi Science movement. We are 1/5 practically of humanity. We don't need to wait for the Western world to accept this. Multiple Nobel recognized. If you come to my talk in the afternoon today, I'm going to talk on cellular physiology, evolution, how cell can work independently. Cyanobacteria, for example, how can it get, or red blood cells, for example, it doesn't have nucleus, it doesn't have mitochondria, but it still survives for 4 months. How can it do? Classical theory is completely a failure in that. So my theory explains how cells can work rather unintelligently, based on simple thermodynamic principles. Alright? So this is going to change, but it can change only if people who come to know about it make some token efforts and give me some kind of a support to advance this further. Otherwise, I have tried to reach out to the governmental machineries, CSAR, ICMR, DST, DBT. It's very slow. Alright? But the good thing is, I could get it published, sir, in decent journals. 3 to 6, 7 impact factor, decent journals. I'm lucky that, that part. So I really hope that Swadeshi Science Congress movement will be looking at my works in greater detail, and if critique my work, cut it left, right, and center. But what I show, the people who are in this field, they told me, you don't shame the community like this. If I don't shame the community is not changing.
Okay.
Yeah, that's the problem.
Superoxide dismutase we visualize as a savior of the plant system, but no, you are
exactly, sir. Sir, I am simple. Ranga, the amount, the context, the time, everything has its place. Wrong amount, wrong place, it screws up. It's like this, sir. My theory, murburn theory, basic gist. It's like this. You take a cloth, dip it in oil, and then set it on fire. The oil burns first. Cloth will burn. So that's what happens to us. That's what murburn is. Right? So you also know photosynthetic, all those cells that are involved, all those plants that have high rate of photosynthesis, they die. Why? Because it is based on wrongs. Collateral damage. There is no other thermodynamic route to get this outcome. The synthetic outcome cannot come without that. The 2-electron process, you cannot get it. Thank you so much. Thank you so much.
Any more discussion?
Any questions? Any questions?
There calculations about how energetic unfeasibility of all the other methods.
Yes, sir.
You didn't give any for your method.
Yes, that's very good question. Madam, your name I wanted to ask you.
No, it's okay.
No, I want to know. I want to know.
That's okay.
No, I want to know. Well, I will show you the slide. That is not a problem. These are my reactions of ATP production, of oxygen generation. You see, very simple methods. It doesn't require. See, these are primitive enzymes. Before affinity-based mechanisms came into being, oxygen is a diradical. It is a diradical. It can easily get reduced. Oxygen gets reduced minus 270 kilojoules per mole. It's the most facile reaction. That's why it became so important for us. Without oxygen, we cannot live for more than a couple of minutes. And this is the reason. So you see these are all bimolecular reactions, madam, and they are highly facile. The other ones are not facile. What happens is that thylakoid, that stacking up, that lipid, why do we have it? Because proton is minimized there. So it enhances the oxygen-centered radicals lifetimes. So that is how the synthesis happens.
And you don't say quantum computing.
There is no quantum computing.
That is not what is supposed to be operating. It's
Yes.
not coherence.
Well, there is, people say there are quantum coherence. There are so many theories there. There are 3 schools of theories. And the point is, they are trying to explain how the randomly distributed light harvesting complexes, how can they give excitons to the reaction center. What I am saying is, they don't need to do that now. The light harvesting complexes generate ox. Now you have one oil to burn in the chamber. There is another oil to നമ്മുടെ എഞ്ചിനിൽ ഉണ്ടാവില്ല സർ, ഒന്ന് കത്തിക്കാനുള്ള ഒരു എണ്ണ, ഒന്ന് ലൂബ്രിക്കേഷൻ. കറക്റ്റ്, ആ ലൂബ്രിക്കേഷൻ എണ്ണ പോലുള്ള എഫക്റ്റ് ആണ് നമ്മുടെ ഈ ലൈറ്റ് ഹാർവെസ്റ്റിംഗ് കോംപ്ലക്സസ് കൊടുക്കുന്നത്. That's how. My theory explains practically everything reported till date. The existing theory, there is nothing it can support. In my paper, there is 50 itemized agenda where I have compared why this theory is right, why that theory is wrong. I have to make the claims first. Then other people will say. My problem is, I don't know. This is the problem that I face. Because when I try to make claim, people expect others to recognize. Others will recognize only if I make claim first. That's what I'm doing. Please, that's what I'm doing. It's very difficult for a person like me to go against the current of 80 years, and I have to go about it like this, man. There is no other way. Thank you so much. Madam, your name you didn't tell. You know my name, Kelath Murali Manoj. It's a good question you asked. It does matter. It does matter. You ask me a question. You don't want to identify yourself. Okay, I'll ask somebody else. Thank you so much, sir. Thank you so much. Thank you so much.
We are wait for a tea and assembled by